cookie to put in your pocket or something for here. We will go through a couple of talks and then we'll have a break where you can grab another cookie or grab some more coffee. And this afternoon's session is really focusing on those new tools and new technologies that we can use to help us deal with some of these double whammy of climate change and invasive species that are going to impact our Adirondack region. And so, since we're going to be talking a lot this afternoon about the alphabet soup of eDNA and RNA and genomics and lots of different things, it might have been a while since you were in AP biology or your biology class right there. And so I volunteered because I'm always complaining to my wife when I'm doing stuff like around the house. I'm trying to fix something or rebuild something, and I'm like, what did my liberal arts degree in biology actually give me? I don't use that thing ever. And so, when we said, can somebody give like a 10-minute kind of like primer on some of these basic concepts we're talking about, I said, here's where I finally get that return on investment. And Dr. Moss and Dr. Shiflett, 20-some years ago, were making me look at these textbooks and, try to figure all this stuff out for me to just go off in the environment and count plants. So this is going to be your Biology 100 quick review. We're not even getting to 101, not much less 200 level. So this is a very, quick overview of some of the concepts that we'll be talking about. And one of the key concepts that we're going to go over is classical cell theory. So it's probably been a while since you thought about when they discovered cells. So we're going to go way back in time to 1655 when Robert Hooke used a new tool, used a microscope. And by using the microscope, he was able to look at a thin slice of bark from a tree and saw all these repeating little patterns. Now these are dead cells, but he was the one who reminded him of the small cells in a monastery, and so that's why they named it after that. So this is 1655. We start to get to the idea that all organisms are made of cells, which is one of the main principles of classical cell biology. The other part is that the cell is the basic building block of life. We are all made of trillions and trillions of cells. These are what's called eukaryotic cells, so they have a nucleus that is containing the DNA and the RNA, and then there's this other surrounded by a plasma membrane on the outside of it. And you can see all these organelles like ribosomes and the endoplasmic reticulum, these things that help us build proteins and do all sorts of important functions. So this is an animal cell. Plant cells look very, very similar, but you'll notice that they have that thick green cell wall that helps provide some of the structures, since plants don't have bones or other different things. This cell wall is actually does a good job of containing a lot of these different parts. And so when we talk about some of our eDNA and picking up for plants, it will be that comes into play. So all organisms are made of cells. The cell is the basic building block of life, and then all cells come from the pre-existing cells that have multiplied. So you're looking up here, looking at mitosis, looking at the cell division that duplicates one cell into two cells through there. So this classical cell theory, we're like at 1850s, 1865 right now. So we're up to the U.S. Civil War. This is the best of the theories, but once again, we get new tools, we get new insights and new research. We build upon the shoulders of giants. We get to stand up there and look out further. So modern cell theory built on it with a couple new features on here. So first, they wanted to say that similar species have similar cells. So That means that the cells that make up you and I are virtually identical in their structure and chemical composition. Likewise, the cells that make up two similar plants, like in this example, two plant species or two fern plants, are very, very similar to them. Cells of like species will be the same from specimen to specimen. And so by having these key characteristics of individual cells, we can use that for identification. The other part is that energy flow occurs within cells, so that the process like photosynthesis, whether they're taking in sunlight in a plant, or cellular respiration, the metabolism and the biogeochemistry of life occurs within cells. And then the other part of modern cell theory is that cells can contain genetic material. So they contain DNA, which is deoxyribonucleic acid, which is kind of the star of it. So on the left-hand side, this double helix structure that is the genetic code or kind of the blueprint of an organism and has these repeating sequences of adenine, guanine, cytosine, and thionine through it. So it's kind of like the star of the show. And then the underrepresented but hardworking understudy is RNA, ribonucleic acid, which has a single strand, and instead of being just located in the nucleus, it can move between the nucleus and the cytoplasm. And these cells, they contain DNA and RNA, which is then passed on to their daughter cells during cell division. So, fun fact, even though we all look very different, 99.9% of our DNA is exactly the same. You actually share about 50% of your genes with plants. And if you took one single strand of DNA in your cells and unspooled it, it would be about 6 feet long. So because you have 25 to 30 trillion cells, they say that you unstrung all the DNA in your inside of you, it would go across the solar system from the sun all the way to Pluto, which is pretty crazy to think about. DNA and RNA work together to create molecules called, generally talked about as protein synthesis. So you can see in there they have messenger RNA can move outside of the nucleus, work with ribosomes, and work with different organelles and produce with things like transfer RNA and ribosomal RNA to be able to produce generally like polypeptides or the building blocks of proteins through there. So DNA and RNA are working together. And then the last thing is that we have genetic variation on there. So through either mutations, so when they are being transcribed, random errors get put it into our codes. And then also a lot of variation can come through sexual reproduction. So you can see these example of having two pairs of genes, but then as they are split across and then recombined, if you look on the far right side, you'll see, over time and generations, you get different variation in the genetic expression, and then that can be expressed differently phenotypically. And that's important because we can have all these plants or animals out there, and they all have different traits. Some of these traits might make just a few of fruit. Some of these will make few fruit, but they'll be very big. Some of these will make a lot of really small fruit. And so then through like selective breeding, we can breed together different plants and try to get the best outcomes of here. And so here's just a kind of very simplified example of showing, over 3 generations how you go from having some plants with a mix of different fruit to having a lot of fruit on there. Same concept we're kind of thinking about with looking at our forests and looking at our trees, trying to find trees and things that are resistant to some of these forest pests, and can we do breeding programs to promote that resistance in there. So so in 8 minutes, that's 350 years of science. It's a very, very quick overview. You can talk to Mark or some of our other much more esteemed scientists in the room about this, but just a quick review of some of the things we're going to be talking about cells and DNA and RNA in here. And so the cool part of it is that science doesn't stop. It's going on. And so we get to stand on the shoulders of geniuses and This afternoon, we are going to be talking about some of these new tools, a lot of them that are using things like DNA and RNA and genomics to be able to help us better monitor and better address the potential impacts of invasive species. So with that, I will give you all an A and stop off, no more biology 100, and we are going to have our first speaker, Aaron. So let's see. Let me get your, where is, oh, ready? Sorry. I'm looking all, nope. Yes. All right. We are thrilled to have Dr. Aaron Malloy here. He is a geneticist with the U.S. Fish and Wildlife Service, and he has come here to all the way from the Northeast Fisheries Center, and he's going to talk a lot about looking at environmental DNA, a tool that we are rapidly implementing across the Adirondack prison.
Thank you, Brian, and thank you for that introduction as well. I certainly remember using that process. All right, so as Brian said, I'm from the Northeast Fisheries Center, which is part of the U.S. Fish and Wildlife Service, located in. The service in general has about 9 genetics labs across the country. Most of them were started as a conservation genetics lab and population genetics and stock management for a hatchery system. But over the last about 10, 12 years, we really began to expand out into the DNA space with a lot more DNA work. So right now, ourselves at the Northeast Fisheries Center, the Midwest Fisheries Center in La Crosse, Wisconsin, that's the genetics lab, and Fish Technology Center in San Marcos. All 3 of those labs are currently doing qPCR work for DNA as well as barcoding. So those are the 2 main analysis techniques that we use for environmental DNA. I'll talk a little bit more about them as we move forward. There's 3 other labs that are doing qPCR work for DNA right now, and that's Warm Springs in Georgia, our Southwest State Aquatic Resource and Recovery Center, and then lab up in Wisconsin. So they're also doing DNA work. The other 3 labs are doing either conservation genetics or, in the case of the lab in Oregon there, that's our U.S. Forensics lab, which is a really unique case. They actually are part of the law enforcement division within the Fish and Wildlife Service, so that's a really cool place to be. Don't know about it, you can check them out online. They go really, really fascinating work for forensics, wildlife forensics. Across those labs, though, we have a lot of expertise within eDNA and a community of practice that have put together a couple of great resources that we'd like to point you to. One is our best management practice guide for doing environmental DNA work. So that's really written for the practitioners, the managers that are out there using the data that comes from eDNA. It's not written for the lab practitioner. So you can follow the QR code. You can find it on our website as well. That's a great resource. You can download the PDF and get acquainted with the information there. We also do a training course at the National Conservation Training Center in Shepherdstown, West Virginia. So that's a week-long kind of hands-on course that we do, both a classroom component and also a field component. Again, really designed and driven for the resource manager or the field practitioner. It's open to everybody, so state agencies have attended in the past, NGOs, we've had graduate students attend, that sort of thing in the past. It's a really great opportunity to get some training, hands-on training, and meet some different people within the space. All right, so with that, we'll get into the nuts and bolts a little bit more. So what exactly is eDNA? Well, that definition has evolved greatly over time, what DNA actually is or what we consider it to be. I've found that over time it's really broad what we consider DNA at this point. I kind of like this simple definition that came from a publication 10 years, 12 years ago. That eDNA is just a complex mixture of DNA from many different organisms found in an environmental sample. There's 2 parts of that that are really important to take home with, and that is it's a mixture of DNA from many different organisms. And that's what makes the DNA sample unique, is the mixture of DNA. So the photo there is from our Green Bay Quico. They're taking some water samples. And You can imagine the sample of water that they're filtering is going to have DNA in it, primarily from bacteria, algae, fungus, maybe some of the invertebrates in the water, fish that are in the water, all the terrestrial inputs, plants, right? An incredible amount of DNA is all mixed into that sample. And most of the time, if we're looking for an invasive species, the fraction of DNA in that sample that we're actually looking for and target, the same minuscule part of the total, right? So that really influences how we treat DNA samples, how we interrogate them, and it also makes us think that it's false. The second part of that definition is it's from an environmental sample, right? That's incredibly broad what an environmental sample might be. And what we see now is an environmental sample could be a lot of different things. Water sample, sediments, feces. People doing air DNA, passively collect DNA that's coming out of the air. Pollinators. You might be looking at the different pollen the insects are visiting, or you might be looking at the insects, which insects are visiting the plant. So one sample might actually answer multiple questions. You might be doing gut content work, or even bulk samples are now considered to be a mixture of organisms. It might be a picnet sample, and then Bailey's trap or insect, something like that. Organisms are all kind of ground up into a single sample that then gets analyzed. All right, so lots of different sample types would be considered DNA. But obviously we collect those samples very differently. We extract the DNA from those samples. Once we get to the analysis post-DNA, things get quite more similar then as far as how we interrogate the sample in the lab. But the collection and extraction process can be quite different. It also influences the way we think about DNA and how we might do our experimental design. So kind of talk about the concepts here of the ecology of the DNA, how that DNA interacts with the environment. So if you just think kind of about these four different examples, right? So air, water, soil, and feces. Think about eDNA transport, how eDNA moves through those different sample media, and how that would influence how we sample and what information is taken. So if you can imagine an air sample, that DNA may have traveled for meters, kilometers, hundreds of kilometers, thousands of kilometers. We're analyzing an air sample for pollen. Newborn pollen exponential as far as the distance that DNA can travel. Water may be less, but we're still probably talking about kilometers of that system. And even what moves around in the soil sample. So if the soil always is rotating, insects are moving and turning that over, DNA definitely moves around in that system as well. Maybe not so much within a fecal sample, but the predator, the herbivore is moving that around. Okay, so go to the ecology of that, how we sample it. If we take a sample of an air sample at ground level, it's going to look very different than it does. So it's very different in the way we think. Same for water samples. Sample surface and sample depth. I'm sticking with this theme of sort of the ecology of DNA. One, it's the sample matrix that influences things, but it's also that physical part, physical environment. So UV exposure, temperature, you have a lot of rainfall, microbial activity, and the water chemistry itself. Those all influence the way the environment, the way it degrades through time, or if it's diluted, or if it's transported from other locations. Chemistry can affect inhibition. That's certain chemistries of water can affect our assays in the lab. They don't perform as well in the lab because there's something that was co-purified in it. So all of these things can sort of begin to accumulate and affect your overall detection probability of that organism. Again, all of these things are concepts that we have to think about when we take a sample from the environment. Okay. The last one I'm going to mention here with the ecology of DNA is just the organisms themselves, right? They do different things out there. They use different habitat, right? So grass carp is going to be doing something very different in that habitat than, say, spiny water breeder is. right? So that natural history of the organism needs to be taken into account. And things like migration, they may only be in the system at certain times of the year to actually detect the DNA, and that's going to affect the sample. Spawning events. Spawning events are great because they release a ton of DNA all at once in the environment, so that might increase your detection probability for something that's really critically hard to detect otherwise. Life stages. Target different life stages, whether it's the benthic stage for our bivalves or the larval phase. And then the DNA shedding rate. All organisms shed DNA differently. Right? So American eel, those things shed an insane amount of DNA. Right? They're slimy, they slough DNA. Detecting eel are no problem. Detecting crayfish, much more challenging. Just do not shed as much DNA as something like this. So as powerful as eDNA is, it isn't always the right tool for the task. So it's always there's variation on your success rate across the board. Okay, so where does that DNA come from? So I have a lot of fish examples. Is primarily what we do at the Northeast Fisheries Center is look for fish. A lot of it's invasive species work, some of it's native species work. But here we're thinking about brook trout. Where's that DNA actually coming from? And it can actually come from a lot of different sources. One of those sources is free DNA. It's cellular respiration. It's DNA that leaks out of an organism, right? It's not actually bound in a cell anymore. It's just free DNA. When it hits the environment, it doesn't actually last that long because it degrades quite quickly. It's not protected by a cell or anything like that. So there's a lot of it that doesn't persist very long. You get a slough of scale and mucus out of carcasses, right? Everything eventually perishes, predation, those sort of things. They can actually leak a lot of DNA into the environment for quite an extended period of time. Because it takes a while for that to decompose and release all of the DNA. The gametes then, during spawning events, like I said, there's a big peak in DNA output during spawning events that can be really useful and built into your experimental design as well. So like when we do freshwater mussel work are difficult to detect. Doing it during spawning season increases our probability of picking up quite quickly. But I would say overall, for something like a fish, the thing that we probably detect the most is feces. There's a lot of the sloughing of epithelial cells from the intestinal lining come out of the feces. That's a constant input of the systems. A lot of what we do detect is mostly feces. All right, so with that, what does an actual DNA detection mean? And for the most part, an DNA detection is just that. You've detected the DNA of that organism in the environment. Doesn't mean you've detected the organism because we're indirectly detecting it. And it doesn't always tell us the status. It doesn't tell us if that was a live organism or a dead organism. Right? DNA is the same. It's going to be detected whether it's live or dead. It doesn't always tell us the origin of that detection. Right? There are inputs of DNA that come into the system from outside of the area that you're working in. So it could be brought in by bird fecal samples. It could be brought in on a boat, bilge water, barge traffic, sewage treatment plants. So there's a lot of different ways that DNA can come into the environment that doesn't mean the organism is actually dead. So that site selection becomes very important. How you pick your sites, limiting and understanding the influx of potential outside. So you can do a lot through the experimental design to eliminate that stuff and increase your confidence that we're going to detect DNA from an organism that is there. Like I said, it doesn't say anything about life stage either. You can detect eggs, larval, juveniles, adults, all in the same way. And we don't get a lot of information regarding the proximity of that detection. So we know DNA moves through the environment. So are we detecting DNA from an organism that was 10 feet away or 2 kilometers away? Right? You can get some indication through, like, based on the sample type and sample matrix and the system that you're working in, but you can't really pinpoint where the organism is. The way DNA moves through the system. Okay, so we'll shift gears here a little bit and just move on to what a generalized DNA would look like. And this is a sort of a water example of filtration. So you got to go out and collect the sample. For the most part, water samples at this point are generally filtered. So there are some cases where the turbidity is too high. There's a centrifugation that's used to concentrate the DNA. But for the most part, it's filtration. After you've filtered the sample or concentrated in some way, you move into the laboratory. That's where we're doing our DNA extraction, using our molecular techniques to detect the DNA. And that's followed by some kind of data analysis. So I get a lot of questions regarding collection. How do I collect the sample? What do I need to collect the sample? How much does it cost to collect the sample? So I put together this slide as just kind of a heuristic model to help you through that process. Collecting an eDNA sample doesn't have to be expensive, but it certainly can be expensive, right? Depending on what type of equipment that you want. So for a couple $100, you can buy a little vacuum pump, vacuum filter, and collect DNA samples. You can spend way more. You can spend up to $8,000, buy a fancy backpack that takes your latitude and longitude, it takes the volume, the head pressure, and all these different parameters. But at the end of the day, you can collect good quality DNA sample with any one of those methods. What's more important is how you treat the sample after it's collected, ensuring you've got cold chain storage, ensuring that it's well desiccated, however that preservation is taken. So there are a lot of different options in the way that you collect the sample. It doesn't necessarily have to be all that fancy. What you're paying for is generally convenience and somewhat of efficiency. All right, so once we've got that sample collected and comes to the lab, the first step is extraction. For the most part, that's pretty boring. A lot of commercial kits are used for the extraction. But the more important part, I think, is the analysis method. How are we actually analyzing the samples and what information we get out of it? So I've broken it down into three basic types. There's quantitative PCR, which you'll hear referred to as qPCR, dPCR, digital PCR. They're all kind of the same thing. The difference is subtle enough. Won't go into it here. Then there's metabarcoding. So metabarcoding is a sequence-based system, and you'll hear that referred to as Illumina, PacBio, next generation sequencing, high throughput sequencing. So a lot of kind of names that's attached to it is metabarcoding. And then I've got point of use. Those are a little bit different. There's LAMP assays, CRISPR assays, a lot of different things going on there. Some of them are qPCR based. But we're going to break it down for each one of these based on a couple of different parameters. One is how many species can we look at. Right? So for quantitative PCR, it's really limited to a single species or maybe a couple. You can multiplex the assays, but things get a little tricky in there. There's some trade-offs with doing that. Most of the eDNA work that we see with qPCR methods is one species. You're targeting a single species at a time. The same is true for point of use. That's generally one species or a couple of species at a time for point of use, which is quite different than metabarcoding. Metabarcoding is really a diversity-based analysis. We can look at a whole suite of organisms or species at the same time. Right? So that's really a diversity-based analysis. Quantitative PCR and point of use are a fluorescence-based detection technology. I'll talk about that a little bit in the next slides, where metabarcoding is sequencing. There's differences in the sensitivity, too. So, like, if you're searching for that invasive species and you want your assays to be as sensitive as possible to the DNA, quantitative PCR is the most sensitive. Metabarcoding is a little less sensitive, but newer sequencing technologies with flow cells, different things that they're doing with the chemistry are improving that as well. So at this point, it's getting close to parity with qPCR. I think what I've seen in our lab is it's probably still a little less sensitive, but it's not that different. Point of use assays, though, are not very sensitive. They're probably 10 times less sensitive than qPCR, right? But they have their place. The main advantage for them is the fact, right? You can do that point of use. So think of yourself as like you're at a port of entry, and you have aquarium shipments coming in to the aquarium trade. And you've got to screen these samples and say if there's any zebra mussel in these moss samples or something like that, right? The DNA all concentrated, the volumes are small. You probably get away with not having an assay that's nearly as sensitive as you would if you were sampling a giant lake or a river system. Right? But what you need in that instance of port of entry is something that's fast. I need an answer now. Right? So that's where those point of use assays really come in and can be quite powerful because you'll get data back sometimes in just a couple hours. Quantitative PCR is pretty quick, but it's still going to take 2 or 3 days. Like that has to go to a lab to be analyzed. You're not doing that. Metabarcoding takes a bit longer. You're probably looking at a week up to 2 weeks for sample analysis. If the labs have absolutely nothing else to do, look at your samples, right? But that's never the case in a lab. Labs are usually overbooked. So it can be kind of a slow turnaround. But sample throughput can be quite high with these, especially qPCR and metabarcoding. Point of use assays tend to be really low, a handful of samples at a time. I think the latest one I've seen are about 8 samples at a time. So you're not going through very many samples in a day. Point of use. And the cost structure is very different too. And so I put some numbers to it because people always want to know the numbers. How much does this cost? It can really vary between labs. When you're contracting with a university lab that's outsourcing that to graduate students, or you're working with a government lab or a private lab. So they can vary quite a bit. I think our prices in our lab kind of fall kind of in the middle of some of these. But anywhere from $80 to $150 for a qPCR sample, and up to maybe $200 or so for that point of use for a bit less. At this point, I'm going to leave point of use behind. I'm not going to talk about it. It is being used to do ENA work for invasive species, but it has a pretty narrow focus on how it's being used. Quantitative PCR and metabarcoding are the two that most. qPCR is the most widely used method, but metabarcoding is being seen there pretty quickly. All right, so quantitative PCR. So this is the nuts and bolts, a little bit more about how an assay actually works. To keep it simple, what we do is we design little bits of DNA called primers, a forward and reverse primer. Those primers are designed specifically to your target organism. So they have to exploit base pair differences between targets and non-targets in the environment. And those are used to direct the PCR process. It's a polymerase chain reaction. It's just the process by which we create copies of DNA. Use those primers to direct that process. We create lots of copies. So in that environmental sample, we might only start with a handful of copies, but after we go through the amplification process, create lots of copies, we might create hundreds of thousands or millions of copies of the DNA that we're looking for. Once we create enough copies, that little probe that's listed there sits in the middle, and it's got a fluorescent label on it. So once we've created enough copies, we can begin to see the fluorescence from that sample, and that's what tells us it's positive. And background fluorescence is higher than our threshold. That's what we're actually detecting in our environmental samples is a fluorescent signal by creating enough copies of it. All right, so that's quantitative PCR in a nutshell. Metabarcoding is a little bit different. It still starts with that same PCR reaction, but now our primers are generic. They're not designed specific to target. They're designed for a generic group of organisms. In this instance, it's fish. So when we go through that amplification process, we're creating copies of DNA that were in our sample of all of the fish. And so because now we've got a mixture of DNA, we can't just use a fluorescent signal. We have to take that to sequencing. So we actually sequence all of those products and get that underlying base pair composition of all those. So what we're really recovering from that environmental sample at that point is DNA sequences. But at this point, we don't know what those sequences represent. It's just a DNA code. So we need to be able to compare the sequences that we take from the environment, or we recover from the environment, and compare that to a reference database of known sequences. And we've built those reference databases based on like bin clips and tissue samples, physical capture. So we know what those sequences are. So by comparing what we find in the environment to that reference database, we can begin assigning looks like here then is just count data. It looks like count data, it is count data. But what we have to remember is what we're counting are DNA sequences. Right? So that's the difference between qPCR and barcoding. So how do we apply that? We can compare and contrast that a little bit here. So quantitative PCR, the main thing with it is the species of interest is predetermined. You have to know what you're looking for because you're designing those assays for a specific organ. Right? So we use that a lot for AIS monitoring, but it's generally used to monitor for something we know is already there. It's an invasion front. It's movement from one area to another. Right? We know what's there. We have a concern about a specific species. That's the context in which we're generally using that. We also use it to do threatened and endangered species work. Distributional status on endangered species. It might be temporal studies for migration, those kind of things. So there's different contexts outside of invasive species, but for invasive species, it's generally something that has to do with invasion from home. We also use it for confirmation testing. What I mean by that is if we get detections with a metabarcoding that are new, we can use a qPCR marker to kind of follow that. When it comes to metabarcoding, the species of interest is undefined. We don't have to know what we're looking for. So when we go through all those horizon scan models and look at watch lists that are created for certain areas, so like I work a lot in the Great Lakes Basin. In the Great Lakes Basin, I think our horizon scans and watch lists have like 50 different fish species. It's not practical for us to go out and search for 50 different species of fish that we're not sure if they're even in a system with a qPCR marker. We would need 50 assays, run that 50 times in the lab, it just doesn't make sense. So when we're broadly monitoring for invasive species, metabarcoding is a great choice because we can kind of target all of those species at once. So we use that for monitoring AIS hotspots, or maybe invasion gateways, where it's a canal system that connects two major systems, those kind of things. Outside of AIS, it's biodiversity assessment, it's diet analysis, right? So those are different use case scenarios. I'll go through a couple of examples of how we're currently using some of these techniques. So this is a sort of an invasion front model here, monitoring, I should say, for northern snakeheads. So this is some work with the ESC Pearson's group. So we know in this instance, this is in New York State down in the Bashkill Marsh. We know there's northern snakehead in the Bashkill Marsh. North of the Bashkill, though, there's access into the Hudson, or the Delaware-Hudson Canal, so you can cross between the Delaware watershed and the Hudson watershed. And there's an interest in keeping the snakehead from making that transition across watersheds. So DEC has erected a temporary barrier in the canal system there at the high point in the canal. I think they're working on a more permanent solution for that. But we've been monitoring below and above that barrier using EDA, looking for the movement of northern snakehead in nature. So I think at this point we do that about 4 times a year. We've been doing that for the last couple of years. But again, there's an issue, we know what's there, we're concerned it's moving, we're actively monitoring that, and there's sort of a plan in place to prevent them from moving. There's a plan in place to maybe put a permanent a solution in place as well. So this is an example of invasion front monitoring we're doing in the St. Lawrence River. So this is a service-based project of the Lake Champlain group. Sorry, this lower Great Lakes Fish and Wildlife Conservation Office. And this is up in Seaman, New York, around in the St. Lawrence there. And the main interest there was tench. So we know the invasion from tench is somewhere in that area. So we are seeing some tench detections up here. But it was about 5 samples out of 137 samples taken over 2 years where we're seeing those tench. That sort of coincides with that invasion front. And that's normally what you would see in an invasion front scenario. It's either you're getting no detections or you only get a couple of detections. Okay? The nice thing about metabarcoding, though, is you're getting more information. So we weren't necessarily looking for 2-dose Goby, but we find 2-dose Goby in that system as too, and they're also invasive. You can see here, tube-nosed goby has a distribution throughout that area that we're sampling, and we're picked up in a much higher percentage of our samples. So, similar example from Lake Champlain. So this is what the Lake Champlain photo now. Here, there's a lot of threats to Lake Champlain. So they're interested in whether snakehead, brown goby, grouse carp, all the big-headed carps, right? So we're not looking for a specific species per se. We're looking for all of those. Fortunately, they're not in the system that we know of, and we haven't picked up any detections. But through the process of doing that work, we're getting data back for tench. We can see that tench DNA is pretty much throughout the Lake Champlain system. European red is also invasive, but it seems to be relegated maybe more to the northern part of the system. So you're getting additional data on all those invasive species, but you're also picking up some stuff with the native species. So in this instance, we're seeing eastern sand darter, which is a stateless fish. So again, that's a conservation focus to some folks as well. And for me, I think that's one of the most powerful things with the metabolic. You're also getting native species. Here's a different application. So this is a diet analysis project. So this is a diet analysis on flathead catfish, which are also invasive species. This is in Edisto River in South Carolina, Barabluff National Fish Hatchery. And what they would experience is they would go out and they would capture these flathead catfish, and their stomachs were eating. There was an interest in, well, what are these things actually feeding on? How are they impacting the native fish? There's nothing in their guts to really identify. So they would send me really nice samples of gut ooze, intestinal ooze, and we'd be then looking at that. And, what we found there was really interesting. So you could look at the intestines, about 60% of the intestinal samples were identified. That number was only about 30%. So like it would typically detect stuff in the guts. And that goes back to that idea of the ecology of our DNA. What's it doing in the sample? How's it being interacted with its physical environment? So obviously in the intestines, it's been in the system longer. There's a lot more degradation. That DNA is just degraded to the point where I'm not really thinking about this. And so it tells me gut samples are a lot better to be working with than intestinal samples. But you start getting a picture too of the native fish community that's hitting. So they certainly hit the sunfish, shiners, and darters pretty hard. That's consistent with some additional years of data. Okay, so that takes care of the examples. One thing I will talk about here, kind of what makes successful EDA. And I think that really comes down to communication. Right? So a lot of you that are thinking about an EDA project, you may be new to it, you don't have a lot of EDA background. Really important to be talking to the labs. We do a lot of consultations with folks that are interested in doing EDA projects, and I would say probably maybe 30% of them we actually work with those folks. A lot of times we just have the consultation, they learn information. Maybe they go to a different lab, they go to a university partner, whatever. It doesn't really matter. You need to have that discussion with the lab somewhere, right? The labs can help you, more clearly define what your question is, help you decide if EDA is even the appropriate tool. It's an incredibly powerful tool, but it's not always the right tool. Choose the appropriate sampling analysis methods. You need to do qPCRs and metabolic. Help with study design, where to sample, when to sample, can we exploit migration, spawning events, different things to help you be more successful with that, with your questions and your goals in mind for the project. And also help develop a budget. So it is probably more expensive than you think. That's always the case with science, but it is more expensive than you think it's going to be. Right? So having that conversation up front is important. But also having conversations on the back end, having a communication plan in place, identifying who your partners are, resource managers, and various stakeholders that might be interested in making sure you have a plan. Collectively deciding what the data interpretation guidelines are. How are you going to determine what a positive actually is? find notification pathways and timelines for that, so that you're all on the same page working towards a common goal. Data sharing expectation. Who gets the data? When do they get it? Who needs to be that communication pathway? Especially with AIS pieces, it can become kind of controversial. The sequence of events of who gets notified. And also understanding what the response options are. It's important to have those criteria in place before you start. Like, what are you going to do if you do get detections? You can go out and get detections, but if you don't have a plan in place for what happens next, is that detection all that valuable? Having a plan in place. All right. And then this one's kind of an outlier here. Just want to mention this. Emergent eDNA technology. eDNA, the space is moving incredibly fast. Technology is moving fast. There's a lot of really exciting stuff going on. There's a lot of research going on, lots of publications coming out. You probably come across it in the literature or even in computer science kind of publications. But I find that it's really kind of overwhelming for folks because it's like there's this need to like always chase the shiny object. And it's all of these things are really cool, they're really great, but a lot of them aren't only that, they're only research and development kind of ideas, right? They're not something that can mature to the point where they're ready for use and monitoring and management. So it's always important to keep that in context. You see that new technology out there, where does it fit on that spectrum? Is it something that's ready for management application, or is it something that's really sort of research and development? That's kind of where our labs come in, being within the Fish and Wildlife Service, Fish Technology Centers. We're kind of in that space of like pulling some of those cool shiny objects and evaluating, determining if there's something that we can use, and if they are, being able to adapt them into that. So we kind of exist in the middle of that research and monitoring. All right, so I will close with this slide. This is just two projects I kind of wanted to point out that are going on. These are really large-scale monitoring projects that are being stood up right now. One is funded through the Great Lakes Restoration Initiative based in the Great Lakes Basin. And this is going to be an AIS monitoring program throughout the basin based on metabolic. So this is monitoring for invasive fish species throughout the basin. So this is going to be a large effort. Ourselves at the Northeast Fisheries Center will be one of the labs that are analyzing these samples along with the genetics lab on Alaska, Wisconsin. So we'll be the two monitoring labs. And it will really be built on collaboration and partnership with other agencies, state agencies, federal agencies, tribes. A lot of different people on the ground collecting samples that will feed into the lab. I think we're in year 2 right now, so we're still developing communication plans. We've taken on a handful of partners on sort of a dry run basis this year. That's going to slowly build out over several years. Full-fledged monitoring. The second one is the Early Detection and Rapid Response National Framework. This is a bipartisan infrastructure funded initiative at the Department of Interior level. So this is a large national effort that is invasive species in general. It's not just eDNA, it's a very large program. But invasive species monitoring using eDNA is a component of that. And that's all based on the qPCR. 3 of our service labs will be monitoring labs that are associated with that effort. So that's our labs in Alaska, Dexter, New Mexico, Georgia, as well as a USGS lab in Idaho will be part of the science support in that process too. But that's another partner-driven initiative where it'll be cooperation with the state agencies and tribes and other non-government organizations that will be on the ground taking samples that can feed into this network analysis labs for. All right, and with that, I'd be happy to take questions.
you talk a little bit more about how does it work? Did you use it for HWA forums, cost, so forth?
I'm sorry, I didn't catch all that.
The autonomous sample. Where would you?
Oh, the autonomous sample.
Yeah. How would it work?
Yeah, so some of those autonomous samplers, that's a great one. That's one of those emergent technology kind of ideas here, right? So this unit here, it's a private company that's doing that. It's based on qPCR, and all the qPCR is actually done on board autonomously. Right? So the samples are taken, DNA is extracted, qPCR happens on board, data analysis is done on board, and through the cellular network, it passes back information to your phone. Right? It's been on their website for about 4 years. It's still not available. So there are a lot of other autonomous samplers that are out there. Part of the EDR national framework is a project in it called ReadyNet, which is using autonomous samplers. In that case, though, it's just an autonomous sample, pulling in samples. Samples are being concentrated, preserved on site, but they still eventually are collected and go to the lab. And those are really great for stream systems, especially for like migratory species, or but they're all point. You're only getting data from a single point. So it doesn't make sense on a lake to only get lots of information from one point. Not necessarily get information across the landscape. So
would it make sense to use it for terrestrial doses, like HWA?
I'm not sure what HWA is. Oh, okay. Did you hear about that this morning? I don't know about an autonomous sampler. They do have little drones that they use and fly around in the treetops with little sticky pads on them, bounce around in the trees, collecting samples off the trees. The sticky pads are then pulled off and analyzed for the eDNA. So something like that might be useful for woolly adelgid. Yeah.
Do you have any experience working with
Yes and no. I haven't been on it. I've been sitting in the box for about 3 years. Haven't had time to actually use it. So NION is another type of sequencing that's done. It works very differently than some of the sequencers that we currently use. It's powerful in the idea that you could actually use an algorithm. The sequencer is, ours is about 4 inches long, 1.5 inches wide. That's the entire sequencer next to a laptop with a USB, and it produces an incredible amount of sequencing data. The sequencing data that it produces has a higher error rate than some of the top models that we currently use. That error rate becomes a problem when we're doing that barcoding. A lot of times it's only 1 or 2 base pairs difference to separate the species. So that can be challenging. With that said, Oxford Nanopore that makes that machine has been increasing the accuracy of their machines considerably over the years. It's getting a lot better than it used to. I haven't really kind of dug into it for a few years to see how good it's gotten. It's getting closer. And it's cheap. That's the other thing. It's a lot cheaper. That sequencer was $1,500 for it. It's a huge leap, and it's an exciting technology to see. I think we'll probably see a lot more.
So one of the big things about DNA is the fact that DNA in one location, you're not getting like patient if it's still actively there. So using it in cases of cases that are down the person. Is it reliable enough for you to say like yes, there are invasives in this area, it's like apple tree or something? Is that what the concern that you have when conducting that kind of research?
Yeah. so basically how reliable is that? So, that's a legitimate question to ask, and sometimes that heating detection can be really hard to determine. So like if you go out and take 300 samples, and one of those samples was positive with a small amount of heat, what is that telling us? That a bit of bird that flew over? Those kind of questions, right? And that's real. In my opinion, you can start to build a line of evidence. So if I see that one positive out of a lot of samples, my instinct is to go back and take some more samples. If I see it again, that begins to build confidence that it just wasn't a random bird. Can I rule out every other thing? Is there a lot of boat traffic there? Is there sewage treatment from upstream? If you can start ruling those things out and building a case for evidence, I think you can really get to a point where you're confident it is actual efficient system of species protection. But sometimes it is an accumulation of evidence. But again, we saw some of those species invasion curves earlier, right? This really is that early detection, trying to get things at the earliest time possible. Sometimes that information is still going to come with a little bit of question. But as early as we can get in that process.
It would be helpful for figuring out where to target.
Yeah, so most of the time we get detections. The first follow-up would be some additional detection work. If we start seeing it consistently, a physical crew going out there to see if they can do something. But it's often used in that context. It's directing field crew.
Are you finding that using AI is helping you refine that kind of data? So if you have some data, AI can go and look at some samples that you can quickly and be able to rule out.
It's possible. We are not using any sort of AI in the data analysis at this point. Again, as those AI tools become more commonplace and more powerful, I think we'll be trying to where AI has a role in what we do. It's not there.
So one more question.
I was curious. What kind of questions can you answer? Specifically,
Yeah, that's a good question. I personally don't do any. The air sampling that I have seen in the recent conferences, a lot of it was desert based. They were looking for mammals and reptiles. They were picking up a lot of mammal signal. It was mostly close to the ground. The higher off the ground they got, the It's a good question. I don't know. Some of it's happened.
Yeah. Well, Aaron, give Aaron a round of applause. Aaron will be around here, so you can catch him at a break if you have any questions. Also, myself, Hannah, who else? We have Chris. We have a lot of people over here that are doing some eDNA sampling. So if you have any questions, you can reach out. There's some resources right here in our own prison. Moving on to our next speaker is Dr. Kurt Kowalski. He is a research and wetland ecologist at the U.S. Geological Survey Great Lakes Science Center based in Ann Arbor, Michigan. His research focused on the restoration and management of Great Lakes coastal wetland habitats, and including development of novel management approaches for the invasive Phragmites that we're going to talk about right now.
Thank
join me in welcoming.
Is there a clicker here? All right, excellent. So I was sitting there thinking about the rest of this afternoon. I recognize, doing the math, I recognize that I'm the third talk after lunch, and I'm keeping you from a break. So wish me luck. No, honestly, thank you everybody for inviting me here. Well, the organizers invited me, but thank you for having me in your community and letting me be able to share some information. Hopefully, this can help you in various ways. And also, thank everybody for their time today to come and listen, learn, and for Carrie. I think that's really important to recognize. So, thank you for all of that. I am hopefully going to tell you a bunch of information about, we'll be telling you a bunch of information about what we're doing with the non-native Phragmites australis in the Great Lakes Basin. And hopefully it will provide you some big things to think about, maybe some ideas, programs to look to implement in this area, as well as I'm going to share some of the technologies, specifically talk about some biotechnology we've been working on, for sure, RNA-based work. I broadened the talk a little bit so that I'm able to offer, I think, a little bit broader perspective on where RNA fits in the broader solutions or efforts that we're trying to work on with Phragmites and other invasive plants. All right, so overall, the story is going to be positive. So this isn't a gloom and doom talk. Phragmites is not a particularly nice plant to work with, but I think there's a lot of opportunities on the horizon, and some of which I'll talk about, some of which I think are still developing. So let me first ask, who has some knowledge of Phragmites australis? You know what I'm talking about right off the bat. Okay, most of the room, that's awesome. How many of you are actively managing it, or manage people who manage it, or programs or something? Okay, so about half the room, awesome. So what I'm really hoping is that this information, this discussion, helps people that raise their hand last, right? The people who are working on the ground in so many ways. My research is hoping to help you, and by sharing this information, helping other people learn and change our daily activities, but then also help with the actual work on it. So hopefully we'll achieve that starting with this discussion today. I, of course, get to stand up here and talk to you, but I'm representing work from a whole lot of people. Very collaborative work that we do with lots of different organizations and agencies, so I'm going to be summing that all together into this talk, so I certainly want to recognize their work, see the names up there, but then also recognize that most of the work you see has been funded by the Great Lakes Restoration Initiative, and also some other funding sources, USGS and others. But so let me dive in and talk a little bit about this plant, and I can do it manually if you want me. Let's see if this is a video of it. Yeah, good. Excellent. All right, so this is Phragmites australis, subspecies australis. And so for the most part, if I just default to saying Phragmites, I'm talking about the invasive subspecies primarily. And as you can see, it is a pretty big plant, right? There are a lot of invasive plants on the landscape. They come from different areas and different regions, but this is one that is definitely has a very robust growth form. You can see the picture on the right. Gentleman standing there, it grows up 15 meters tall, 5 meters, 15 feet, get my units correct here. And it's up to 2/3 of the plant is below ground. Biomass of the plant is below ground. So I'll show you pictures later. So there's a whole lot of below ground biomass that's going on. As you saw from my little video there, big back, moving forward. Right, so here are some of the characteristics that really make it so aggressive and outcompete native plants in such a robust way. Again, very tall. It has this large fluorescence you saw at the top, right, from the image. So there's a lot of seeds that produces. There's some work that suggests that there's variable seed viability. That's kind of a separate discussion, but it creates a whole lot of seeds. It has these wide leaves, so it shades out other plants. It grows very quickly. It grows earlier in the spring. And again, has extensive underground rhizome network that it spreads also vegetatively through these rhizomes and root systems that go underground. So there are multiple haplotypes or genotypes of Phragmites around the world, and the one that we're wrestling with in North America, ones that you have in the park, I saw in the thruway on the way here. I understand you have in the park. I took a walk down by the river. I didn't see any, which is a great thing, but I do understand, it's in the park. And clearly on the east coast of New York and along the eastern seaboard is very much a problem. So focus less on the green. You can't put green laser, say here, on a green blob. It doesn't show up. So don't pay attention to the points as much as the blob. So it's the green that is the Eurasian haplotype, European haplotype. The plants come from Eurasia primarily, and that's the non-native version that we have for the most part here in the northern U.S. There's some different haplotypes down in Louisiana and some of the southern states, but that's the one we have for the most part. So then also have a native, and I honestly don't know how much you have if you have native within the park. There's a little bit of native in the park. I am from Michigan, Ann Arbor area. The native is all over the place, like patches, so not like large patches, but once you know you're looking for it, so it's quite common in our area. So the point is, the native is still on the landscape, and it's not a one-for-one where the invasive outcompetes the native. They occur in similar habitat, but not necessarily a one-for-one kind of thing. And I always just talk about the native that it just plays well with other plants. I'm a wetland ecologist, so you'll see a lot of wetland pictures in here, and so it just doesn't outcompete the the other native plants like the invasive plants. So there's some phenotypic ways to tell by looking at the characteristics of the plant to tell whether it's native, or the native or the non-native. The native oftentimes has red stems. You can see generally lower on the plants. It's not nearly as aggressive. It has a little. I'm not liking the green laser. But it's a little less dense seed head, right? So it's a little more sparse, a little spread out, a little less aggressive kind of thing. And then bottom right, you can see the top leaf there is the non-native. It's subjective, everybody's eyes are different, but it's generally a little more of a turquoise, a little more of a blue-green versus the green, and it's the native is less robust. So there's ways to tell the difference in the field, but honestly, you really need to do a genetic test. You need to do send it away to get some qPCR or other work done to really tell where it's from. And I'll give you a way to do that. You can get it for $20 or $30. Generally, you can send samples and find out then which it is. So ultimately, you can do some rough calculations in the field, but you really want to know by testing genetically. So for those of you that haven't seen this level of Phragmites and what it can do, let me show you some meta pictures. Right? This is what we have in the Great Lakes, and it's all on east of all of the marine coasts. So the Pacific, Puget Sound, Chesapeake Bay, Gulf of Mexico, all over the place. And it has all these impacts that affect us, biodiversity, outcompeting native plants, reducing overall biodiversity. If you're a turtle or a herp or something, it's pretty tough to move through here. Deer move through, but it reduces bird habitat. There's all kinds of impact on wildlife and fish, areas where that's letting out the fish to get through. Also impacting nutrient dynamics and the flow of water. So the Departments of Transportation are very interested in this plant because you're trying to keep water or roadways clear, intersections, so views get impacted by this. So DOTs are very much interested in this plant as well. And if you're trying to keep a ditch open, a farmer or something, or trying to get water conveying, this is challenging. It also impacts people, impacts all of us, right? So the picture in the upper right-hand corner, that's on, I believe, Lake Michigan. That's a 2-story house, and that's a beach. So that is your beach. That's probably not how you would want it to look. Even if you didn't know what Phragmites was, if you wanted to use this for your kids or grandkids, it's not working as a beach. So it's challenged that way. It's also a fire hazard. So oftentimes where it grows in and around houses when it's dry during the wintertime, it's very much a fire hazard, which means it's important to people as well. So all Lakes, these together are part of the reason that it's on the radar for a lot of people in the nation. You see the end maps up in the upper left-hand corner, each of those green blobs. I think this is by county. I think I did there. The point is there's wide distribution across the U.S. and Canada. We work closely with our Canadian counterparts. And the bottom is just a separate data source, again, to show you that this is really a North American problem. So as you're thinking about it in the Adirondacks or surrounding communities, it's the same plant, the same general challenges that are being wrestled with really all over North America. And that's why it appears in so many different management plans. So again, I mentioned the Great Lakes Restoration Initiative, but in the Great Lakes Water Quality Agreement is a big deal that we all work around, sharing water binationally, multinationally actually. And the rest of these don't matter. The point is it's in plans, right? So it's in most of our various organizations, strategic plans in some of the invasive species or fragmites specifically. So it's a very large scale issue, very intractable in the size, you saw the plant, you saw the distribution and so on. So let me introduce this idea of collective impact, because that's really a driver concept that we've used every time to really help with this issue. So collective impact is a thing, like you can Google it, and there's papers out there on this idea. But it's really setting out a formal framework to make a collaboration that ultimately gets all of the work going on the landscape or for whatever issue you're working on, gets everybody working towards that same goal in a very concrete kind of way. So there are these 5 tenets. So you have to have a common agenda. Everybody has to be focused on the same thing and agree on it. You have to have shared measurements, so everybody's going to agree. What does progress mean? How are we actually moving forward with this? We have to have activities that build upon one another. People have been collaborating forever, right? But it's not just not bumping into one another. It's building in an additive way towards this common agenda, towards this common goal that we're working on. And I would say also that good communication is always important, but the backbone support is part of what sets this idea of collective impact separately, because you need somebody thriving, right? We're all busy. We're all really busy. And so having somebody to maintain the communication, set meetings, and help develop a common agenda, and write things out, and coordinate, you need to have a group or somebody play that role. That's pretty important. So Kenny and Kramer, 2011, put this out at Social Science World, thinking about child obesity and things like that. So these really big intractable issues. We took this idea, again, and applied it to Phragmites. So this is just my graphic. Again, it's not just not bumping into one another, but it's working in an additive way. So we applied this concept and modified it for Phragmites. We have this paper that came out in 2015, and I'm happy to share. I'm going to show a few references. I'm happy to share them later with the group. You don't find them yourself. But this has been a guiding force in many ways of how in the Great Lakes we've been thinking about Phragmites and developing science, and thinking about, again, all this within the context of climate change, and thinking about how the variables and stressors that we know and are working with now, it's a moving target. So we're going to have to continue to work collaboratively to move forward with our individual priorities. So part of what that paper laid out and where we've been going here is we helped create what ended up being the Great Lakes Phragmites Collaborative. And so there's a website down there at the bottom that is, again, we're focused on the Great Lakes, but it's the same plan. So you'll find that there are a lot of resources there that we think are going to be very helpful to your work with Phragmites. This collaborative is the group that's working towards a landscape that's not impacted by the non-native Phragmites. And so part of the collaborative said, hey, we need a website. So I want to give you the impression the website is not the collaborative. The website is just the easiest way for all of us to see the case studies, see what a landowner does for managing Phragmites, see what a land agency does if you have water, if you don't have water. Connect with your local PRISM here, or CISMA, the Cooperative Invasive Species Management Agency. I'm getting a nod, yes. Right? CISMA, they used to be Cooperative Weed Management Agency. They're
your Michigan version of PRISM.
PRISM, yeah. So just a different version, different names, acronyms for different ways for people to organize geographically and combine resources and really work together on local issues. There's science work up there. There's the maps. There's a really cool newsletter bottom right-hand corner there that has we have some very creative people creating cartoons through time that are like science through kind of fun. The rest of your family will like it. So a lot of resources here I encourage you to go and check out. Even though it's Great Lakes, I think it could be pretty useful to you. And this whole idea of a collaborative, I think, can be useful to think about. There may have some, maybe a space within what all of you are working on that something like that could be useful. Oh, it is. So the APIP work is on.
Well, it does seem like we're not part of it. Actually, everyone should know that the western part of the Adirondacks is part of the Great Lakes Basin.
Great. Thank you.
We are part of the basin.
Awesome. So let me talk a little bit about, these are the current approaches for managing fragmentation. These are the standard tools and levers that we have on hand. You can herbicide, Invasive, glyphosate, combinations of those. Generally, if you choose herbicide, that is very effective. You know herbicides can work, right? There's mechanical, so you can crush it, you can cut it. That generally makes you feel good, but it just comes right back up. Prescribed burn in certain situations, get rid of that biomass, or if you have control of the water levels, a dike wetland or something like that, flooding and water can be very effective as well. And these are, of course, used in combination as well, depending on the situation, depending on the tools we have at hand, funding, all of that. But these are the tools that we currently have available. Part of the challenge is, though, that there's variable effectiveness with these, and it's not always clear which choice is the best choice. So if you have, some of you have a long career in plant management, and others maybe say, yeah, I've got this plant at my cottage or something. What do I do about it? So there's lots of different entry points. So how do you know which approach is the best? That's a question that we're working on. But there's also the realities that most of these are not species specific, right? If you flood an area, you're impacting all the species, plant species that are there. If you burn it, you have that broad impact, right? So you're not targeting just one plant. Herbicide, you do hand swipes, you do like one by one for sure, but that doesn't work if you have hectares of area to work on. So the other thing I point out here is, and it came up earlier, is experts in science, we know some, right? But we don't have all the answers. And if you ask experts the best way to manage Phragmites, you're going to get a bunch of different answers. So there's expert disagreement. So even though we do know how some of these things work, there's some disagreement and some learning that continues to go on. Which is why the Collaborative and USGS and a whole bunch of other partners with Great Lakes Commission help stand up the Phragmites Adaptive Management Framework. And so this is a formal adaptive management program that's focused on reducing that uncertainty about which treatment works best, given your site conditions. So it's beyond trial and error. It's an intentional way to reduce that uncertainty. And it also is an effort to learn from all of that action that's going on in the landscape. So, top graphic, people are doing things all over the place, all over the place. This is a way for that information, those data, then to help train a model, we have state transition models, so on, produce guidance back to people as part of the program. And then they do something, hopefully follow the guidance, maybe not. Monitor again, we just learn something, right? So producing data-driven best management practices is what this is doing. It's been in play for now 5 years, roughly. And it's really been great to work with the Great Lakes Commission to help create graphics like this, so you can go online and see a dashboard about the results that the model is producing and where the action is happening and all that kind of thing. But this is working with the people and communities, and all of the managers are already doing something on the landscape to learn from what they're doing, to do better science, and to do better management in the long term. So again, this is notable that it's a sub-page from the Phragmites Collaborative. You can see the URL there down at the bottom. So this is ultimately getting back to which of these treatments works best, right? Well, these are all conventional treatments. We're familiar with these. There are limitations. Maybe you can control water levels, maybe you can't. maybe you don't want to use herbicide. Maybe you're working in Canada where they can't use herbicide over water. There's all kinds of challenges with our current tools, right? So overriding messages was that we need more tools. We need more ways to try to tackle this plant and a lot of other invasive plants as well. And so that started off a number of years of work. I'm going to kind of summarize here in the next set of slides, focusing on some new approaches that we've been working on with a whole lot of partners. Now, the upper left-hand corner, that's a moth. That's insect biocontrol. I'm not doing any of that. I really don't know much about that. But I want to recognize that it is a treatment and approach that is being worked on. There's been a lot of work at Cornell and elsewhere for engineers to look at natural predators in their native range and think about which of those are here. Might we cultivate those insects and use them as a biological control against Phragmites? And that's still working its way through the regulatory process here in the US. But on the Canadian side, they are starting to test out in the field and see if they can develop the laws and get them out into the field in right numbers, and then see if there's ultimately impact on the plants themselves. And so there are, again, back to the Phragmites Collaborative website, there's been webinars and so on already on this. Michael McCavish at Master University and others have talked about this one. So you want to know more about that? I'm not the right person, but I want to recognize that it's out there. I do get to talk to you about there's two research areas that I and a whole lot of collaborators have been working on. First is working on the microbiome, and bacteria and fungi, the role that they play in invasive plants, and then targeting plant traits. And that's the RNA work. So I'm going to touch on both of these because they think they can work in conjunction, and they also are part of this suite of new tools that we're working on for this plant. So let me just first start off by talking about the microbiome a bit. So, if you think about probiotics and what we know about the human microbiome, think about human health and digestion, I think we probably have a pretty good sense of how important bacteria and fungi are in the human body and our human health. They're all over us, in us, around us, and it's pretty well established that they provide a lot of positive benefits in certain ways and when they're in balance and so on. It's the same story with plants. So we can find all kinds of microbes on the plant surface, in the cells, in the roots, in the rhizosphere, the root soil interface, all over the place and all over in the soil. So same story, they are very well known to provide benefits to plants as well, just like humans. So again, it's been pretty well documented. This is just a paper in Science that I pulled out. Heat tolerance, stress tolerance, those types of things related to microbes. So our core question is then, can we screw up that relationship? Right? Can we target that relationship, that symbiotic relationship between the microbes and the plant as a form of control? So this work has been, again, with Rutgers University primarily, but also with Indiana, like Keith Clay, and then moved to Lane, so you see another logo up there, and also at the University of Michigan. That we took this idea and said, all right, why not the scientific community? We've got this Phragmites collaborative going on, which is about the people, information, and action. It's about coordination. Can we tweak that and make a collective or a collaborative, take a collaborative approach to into the research world? So that's what we did, created this multi-institution collaborative to really focus on this relationship between microbes and the plant, and not just study it, but also figure out, can we use that as a management tool? Can we turn that into something that's helpful to make management decisions on the landscape? We've been working on this and working together on this for a number of years. There's a bunch of papers that we've been able to put out that have help sort out inventory of what microbes are on and around Phragmite target, what they do. And that's also led to some development of like, all right, what treatments can we have, or what treatments can we develop to try to target those microbes? And so, worked with Jim White at Rutgers University. We've actually worked on a patent application for a non-toxic bioherbicide that I'll talk about in just a second here. But that group, that symbiosis collaborative is ultimately what we call it. Big group of people, voluntary, who wants to be part of this. Let's all coordinate, lots of meetings, talking, and so on. and we're able to put out this paper that laid out a research agenda. The research agenda, again, is to lay a foundation, a framework, so that those of us in the research community don't compete with one another for things. We're working together because everybody has their own area of expertise and priorities and lanes and so on. So anyway, this has been very successful to help move along the science and also towards a microbial-based bioherbicide. So we've been working on this for a number of years, and those are the basic components you see here. These are all non-toxic things that you would find around, sometimes in human health, sometimes around a citric oil. You need to be able to penetrate the leaf cuticle. It's always a big challenge, just getting into a plant something for invasive carps or something that can eat something. Plants don't eat. We talked about it's tough to get into plants and working with plants. And then there's just different components to allow the bioherbicide, I think the liquid, to stick to the leaf. So glycerol does. Arginine to really stimulate the development or the generation of ethylene. And ethylene then breaks down microbes, and so that's the connection to the microbial community. And then some stimulants of sucrose for those microbes to be working on. So we've been working through what this cocktail would look like, what the composition of this bioherbicide would look like. And so this is the general process that we kind of have to test of like, all right, here's what we know these different components are, figure out the right measurements and the right balance of them to stimulate that microbial response, treat them, greenhouse, treat out in the field, monitor plant response, how well are each of them doing, and then analyze and go back again. And the challenge with plants is that we only have one growing season. Our growing season in Michigan is about like yours here, so it's slow. Kind of out in the field, it's year by year. So we have to think strategically about which questions we're asking related to how frequently we have to apply it, how much, what are the composition, all those types of things. But I can show you some dramatic, cool pictures that it's working. So like this is what we get. We're hammering the plant within days or within a week. You see here, Phragmites is very brown, withering up. The leaves are cupping. It's not a happy plant. So here's some full control on the left. We're working 1 meter square plots. This is the image on the left. This is our control. And the reason it looks very open is because we started by cutting all the biomass so that it was logistically easier, and then also the plants were smaller. Just again, logistically to be able to apply an herbicide liquid. But then we did some treating every 2 weeks. It's in the middle frame, and then treating every 4 weeks for the summer. So dramatically, in a good way, right? Brown, we're trying to get rid of this plant. But the challenge is that unlike glyphosate or some of the other here, it's not going systemic yet. So it's not going into the roots like we wanted it to and thought it would to disrupt the microbe below. So we are continuing to, why that is the case, and also looking at more direct application of bioherbicide to the plant roots. That's really where the action is, and where the microbes and plants are interacting related to nutrients. And also through time, we're able to look at, all right, maybe these, the plant is coming back, but underground it's really hurting because all of those underground resources means that it just has a huge tank underneath. So I just keep putting up resources, and we hit it, puts up more resources, and hit it again. Eventually the tank is empty. Just like if you were to mow it 7 years straight, the paper out for 5 or 7 years straight, okay, you're going to kill the plant because it goes up, there's nothing more, nothing more, nothing more. So, we're working to figure out more longer term what we're doing to that underground biomass, what that relationship is with the microbes. So, let me move on then to RNA, because I know that was like the main target, but I thought it useful to share those other points because that's the broader context as we think about targeting plant traits. Using RNA to target plant traits. That relates to how the management community what tools the management community wants, how things are being applied, as well as the microbiome, and thinking about how the microbiome ties in with the work that I'm going to be talking about here. So the next set of slides here this is work, again, mostly by the Corps of Engineers, the Verdict Office in Vicksburg, Mississippi, but then also Wayne State and some other partners. So Aaron gave us a good overview. Well, actually, first Ryan gave us a good overview of RNA, DNA, big picture kinds of things. I did remember some things as well. And then Aaron did talk about the DNA. And I'm going to talk about the RNA side of things, and it goes back to the traits, right? So if you think about the traits that Phragmites has that makes it so big and aggressive and dominant, outcompeting native plants, because ultimately that's what it's doing, right? It's outcompeting the native plants. So it has this huge growth form, lots of stems, lots of that underground biomass that's creating these big rhizomes that grow throughout the system, and the seed output. Well, each one of these traits, of course, has a genetic basis, as we heard. So the question is, can we shut those off? Can we stop the expression of those traits and make the plant less competitive? So that's really what we've been focused on with this work. And it comes back to genomics. It comes back to knowing what is the roadmap for all of those traits. What is the genome for the non-native Phragmites and ultimately the native Phragmites? It was not sequenced until we did some work with LSU and others, and were able to publish that a couple of years ago, but that's the roadmap, right? So now we have this plant that is sequenced and described, and so now we can start to pick out the traits from that roadmap and figure out, all right, what do we want to target? And also with that is the real question about the native. How does the non-native differ from the native? So in the process of doing similar genomic work with the native plant, to look at those comparisons so that we understand how much do they differ. And as we're picking genetic targets for the work, being thoughtful about the native Phragmites and ultimately other species as well. And so that's what's really helping guide the targets we're doing. But that first step goes back to knowing that genetic roadmap through the genome. So what we're focused on is this gene silencing or limiting the expression of certain traits. And foundationally, this is not the same as gene drives or CRISPR DNA-based treatments, because this is focused on the RNA. So the DNA is persistent through time and among generations. The RNA is a transcription of the DNA. So it's by generation. So it's not transient. The RNA is not passed on from generation to generation. And so we're focused on that, blocking the expression of the traits and thinking about the traits that are going to be most damaging to the plant, right? Photosynthesis, growth, things like that. But then also, as I'll touch on at the end, thinking about what traits are going to change in the context of climate change. And maybe it's warming, and maybe it's more severe storm events, or whatever it might be. There could be some opportunities there as well. And then, as we're testing this out, figure out, is it actually making Phrag less competitive? For sure, if it's dead, but what if we just minimize its growth, or change its growth pattern, or when it's growing, and those types of things. So overall, this is what we're really focused on. So here's my little schematic. It's a cartoon I like to show to provide just this baseline understanding of what we're trying to do. So again, you have the DNA, right? So that is the roadmap. That's holds all the keys. That gets transcribed. This is the normal process. Gets transcribed into a messenger RNA. That gets translated into proteins that result in the traits that we see. So the plant up here is big plant, rhizomes, and that type of thing. So going backwards. All right, so we want to hit those traits, we got to go back. So if we don't want the traits, we want to target the protein. We're going back to this post-transcriptional space of targeting that messenger RNA, that single-stranded RNA. And so this is leveraging a natural defense mechanism called RNA interference. And it's well published, it's well established, and it's in plants and lots of other organisms, actually. And it's this process by which it was actually first described here in 1990 in this paper as they were studying petunias. So this is petunia. You can tell, right? So this is a purple petunia. And they were thinking, all right, if we made a more purple petunia, better, more money, right, more whatever. So they added a messenger RNA coding for purple. Just call it the make purple double-stranded RNA here, actually. And it came out white. They're like, what? Like, no, wait, what's going on here? Well, ultimately, that's where they figured out this process of RNA interference, where the cell detects and plant detects suspicious double-stranded RNA, it's going to degrade that and the single-stranded RNA that goes along with it. And they actually, this process was formally described and got Andrew Fire and Craig Mello got credit for it in 2006 for the Nobel Prize in physiology in medicine. So going back to my little graphic here, what does this mean? So if we were just to say that there is a gene silencing vector for double-stranded RNA, let's call it the photosynthesized vector. So let's add the photosynthesized double-stranded RNA in there. Plant sends out these isoproteins, says, well, wait a minute, no, we're going to get rid of that. That gets rid of the messenger RNA, proteins, and then the traits. And then you don't get expression of the traits. That's supposed to be a little yellow, right? So ultimately, by using this process, we get this species-specific, because we're targeting a very small set of code, genetic code, and it's non-heritable because it's RNA-based. Because again, the DNA is still sitting there and getting transformed on to the next generation. So conceptually, it's like glyphosate or an herbicide where you're treating a generation and it has an impact, and then you kill the plant, then I guess there's nothing to transfer on to the next generation. But if you don't, then the DNA is still there, unaltered and untouched. So that's why it's not heritable, unlike a gene drive or something like that targets the DNA of a population. So this has already been in play in lots of different ways, and I'm sure there are more. These examples are, I think, probably a year old already or something like that, but it's already been used in human health. There are examples of RNA interference being used in human therapeutics. And actually, just I don't know if anybody else noticed it, but there was the Nobel Prizes for medicine again were awarded earlier this week. There was one for microRNA, miRNA, and it's closely related to mRNA. It was a big deal, and they got the prize because they're thinking about the human therapeutics. And part of what miRNA does, best I can understand as a wet biologist, is it regulates the amount of the trait expression. And so it's not just tying. I showed messenger mRNA versus miRNA, different types, little different roles, but closely related. I thought it was pretty cool timing when we talked to you today. So another Nobel Prize for this similar type of technology. This being used in human health, also used in agriculture. Arctic apples are a product that are out there, and they're using RNA interference to limit the browning of these apples so that they stay non-brown. So they don't brown.
Non-brown.
But also used in the invasive species world as well, or the pest world as well. There's a product out there that just came out, I believe, last year, put out by Green Light Biosciences. And this is a product that it uses lead prona as the active ingredient in a product that they put out for the potato beetle. And so the idea is that then this has the RNA code for the insect. So when the insect then feeds, then the insect dies, and nothing else is affected because it's just targeting that potato beetle that is herbivorizing the potato plant. So it's out there, and there's a regulatory process currently in place here in the U.S. to move forward with these types of treatments. So as we're envisioning this, we're not targeting insects or other pests, we're targeting plants. So we're thinking of this as a spray, thinking of this like as a liquid, and we have to talk about those targets, the gene silencing agents we call them. So you have that genetic target, you also have to get in the plant. And that's been a real challenge, so both identifying the targets, but also getting it into the plant cells. Because the other part of the cell wall, the green you showed, that's an incredible defense mechanism. Plants are evolved not to let anything into the cells. And so nanoparticles, carbon dots, nanotubes, there's a pretty good body of science out there as potential carriers to get past that nucleus, that cell wall in the nucleus, and carry the gene silencing agents, that little bit of RNA, into the plant cells to have their impact. That's my little graphics to get past the cell wall and into that cellular machinery to do its work of limiting the expression of the certain traits. So this is most of this work is going on at the ERDC, the Engineering Development Research Center. It's part of Engineer's Office in Vicksburg, Mississippi. And so we have identified and already show limited expression of certain traits, right? So we can show this in the lab. We can measure this in the lab that we are getting in the plant cell and we are limiting the expression of certain traits. Challenge has been getting this to show phenotypically, so within the plants themselves. You see the bottom right, you see some leaves and withering and so on. You'd love to see brown, right? Completely yellow or not. But plants are really tricky. And plants have multiple ways of completing processes, photosynthesis and other processes that we are targeting. So we are continuing to work in the laboratory setting and looking to move to the greenhouse and ultimately out in the field, hopefully within a year or two. And so just summarizing this work up, we're finding that nanoparticles and some of these other carriers are showing effectiveness to help getting these RNA-based treatments into the plant cells. We're seeing reduced expression, so it's working, but it isn't there yet, right? Because we're not seeing it in the plants to the degree that we really need to see. So we're continuing to work on that process, both gene silencing agents, getting it in, and then how much, how often, all of those types of things are downstream problems. We're still working on that initial science. So just a couple more slides to finish up here. So again, I didn't talk about climate change, but everything you just heard with this talk, you think about adaptations through time as conditions are changing. We are actively working with the RNAi work to think about, all right, so especially in the Great Lakes, we think about flooding, expecting different water levels, different amounts of water, different times of the year. Well, are there certain traits for Phragmites we might target that make it more vulnerable to flooding? Are there traits we could target to make it more vulnerable to drought or temperature? And I don't have a good snow picture, but I have the frozen frag picture up there. And limit its ability, bottom right, those are runners, those are storms. That's less than one growing season. So you can see it just, it's advancing fun on this mud flat coastal wetland in Lake Erie. But as water levels change and so on for us in the Great Lakes, might we target certain traits differently because of those conditions? So we're working on that. And then I guess I'll finish with this and just say that what we're doing is focused on Phragmites, but we're doing this in context of lots of other plants. I'm thinking about, we've already started working about European frogbit, Hydrochirus morsis-rene, which is another growing challenge in the lower lakes, also maybe here in the park. Nice, cute little floating leaf, like most invasives. It's not Phragmites, but a lot of invasives, right? They're very pretty. It's a very small little floating leaf plant that just creates a mat of vegetation that ducks and organelles really don't like. So we're thinking about other plants in the wetland world, but we're also thinking it's going to be a small pivot once we sort this out to tamarisk or cheatgrass or kudzu or hydrilla. Think of submerged aquatic plants. We're not there yet, but that's going to be on the radar. Probably not my career, but that's going to be out there. So I had the privilege a couple weeks ago. I went to Australia and talked to the CSIRO folks because they also are working on RNAi. This is really cutting edge stuff. They have a different suite of invasive plants, but we're all banging our heads against similar problems. So we're developing collaborations and really starting to develop more formal partnerships with them as well to be able to share science and work towards this collaborative approach of developing the science and hopefully getting more tools in the toolbox for managers and outliers. So with that, I want to get my email down at the bottom there. I'm happy. I think we have a little time for questions now. I'll be around the rest of the day. Email me later. Happy to talk more about this. The Bright Cloud website. I and others have talked about this technology more, so you go and check out those resources. And ultimately, I'm optimistic. I think they're going to be these new tools are going to be. They're not going to solve everything. They're not a silver bullet, but I think they're going to have a place within the arsenal that the managers have. So, thank you. Thanks again for having me. Yeah, the question was, is bioherbicide and the pictures I showed impacting the non-phragmites plants? Yes, it does not seem to be phragmites specific at this point. We're not sure if there's, phragmites is a grass, so maybe it's going to have selectivity towards grasses, impacting more than the broadleaves, but we don't know at this point, other than it seems to be more of a broad scale of impacting machinery and all plants. Thanks. Yeah.
It's the feeling that there aren't interference gases making like tops. Is everything sit kind of 2/3 below?
Yeah, thanks. So the RNA interference work is focused on expression of traits within the plant, which apply throughout the plant. So we're topically applying it now, but there's evidence that it does move systemically, and that the plant, so we could apply topically and have it impact the whole plant. So glyphosate gets carried down, we apply it all, and the plant is translocating all resources and have maximum impact. And we envision that these treatments will be similar systemic movement and then hit the below ground resources much above ground. But so far we haven't gone to answer that question of does it make more sense to apply it underground or not? We don't know. We haven't explored it yet. Thanks.
Could you add Japanese knotweed to your list? Is that rhizome different?
Yeah, Japanese knotweed is a problem, and the reality is, depending where you are in the country and the world, there's all kinds of really nasty species out there. We worked, other people in our office worked on Rainbow Liver Sweet, a whole bunch, and so we've had discussions about who and how, but the reality is of all of our work is you only have so much money and so much time, and so we're focused intently on phragmites with our head up, and recognizing there's a lot of others, and the hope is that we're doing this in a way that makes some progress with phragmites, but then others can pivot and work to all the other species.
So in human gene therapy, they talk about viruses as vectors.
Yeah.
Are there any viral vectors?
Yeah, so the question is in human therapies, viral vectors are used and thought about for delivering products at cellular level. And the answer for phragmites is yes. There's been work on using that. I mentioned nanomaterials as potential vectors. There's also work been done. We're using a disabled virus to get it into the plant. And there's pros and cons with each approach. But to answer your question, yes, that is something that's been thought about and has been worked on. Maybe part of the ultimate solution. Thanks for that. Go ahead. Yeah.
Is there enough known about the basic biology of rhizome formation to identify genes sensitive to driving their expansion, their establishment, that like those be a target for these kinds of approaches, especially the microRNAs that we've heard so much about this week.
Yeah.
They shut down and control not just one gene, but the whole set.
Thank you. Yeah. So targeting the rhizomes and that underground growth makes a lot of sense, and that is where we want to go. So we want to hit all of these characteristics that are going to have the most impact on the plant. We have the genomic work is just a couple of years old, and the work that was already in motion before we had the full genome. Work was in motion was targeting some of the photosynthesis genes and some of the growth processes. So we haven't targeted the rhizomes directly, but to me it seems very not only reasonable but wise to target those. And I envision a cocktail. Once we work out these individual pieces, but we would want to have a cocktail that targets multiple aspects of the plant. If you're going to go out and do the physical application of the treatment, maximize the targets for that treatment. Thank you.
Targeting photosynthesis.
It is. True. Yes. Yeah. Thank you.
Can you comment on a timeline for the development of this technology in comparison to like biocontrols? Typically with bio's going
Yeah. So the timing of what we're working on, clearly we're still very early stages of the work, and the realities of taking what we are describing here and whatever endpoint we're able to get to go from there to a product at the hardware store or at Lowe's or something. That's a long path. And clearly, there's so many hurdles from scaling up and cost efficiency and the regulatory, reality risk assessments, right, to make sure that it's only targeting what we think we're targeting and how long RNA is unstable. We heard that earlier. We know that, and that's actually a challenge for us because if you're trying to apply it to a plant for it to all the way get in, it's degrading before it gets all the way into the plant. So, the longevity of or it lasting too long is not as much of a concern as it lasting too short. But to get your point, there are a lot of things to sort out before it is a tool that's useful or useful and available to lots of folks. Years, yep. And and that's, I guess I would say, for the biopesticide RNA work, the bioherbicide work. There are already active discussions with private companies to figure out ways to partner, and there's interest in trying to leverage their resources and our resources at Rutgers, USGS. The insect biocontrol work, that's, as I mentioned, is already in the field in Canada, and unclear the status here in the U.S. So that is already in motion. Insect side of things. Thank you.
I can just answer that question, but so I saw bioherbicide with patent. So is this something USGS themselves try to bring to market, or just sell the rights to? Like, how would you get into the hands of practitioners?
Yeah, great question. So you mentioned seeing the patent and how would USGS help get it into, that is still being sorted out because we're a public agency, federal agency, so our job is try to get these technologies and figure out the source, figure out the science to get it, to move the science forward. And at the same time, we're encouraged to work with partners in patent ideas to make sure that we're helping put the formal bounds on the intellectual property that we have and go through the processes to move that forward. So it's really unclear how that's going to move forward. Well, I will be doing everything I can to make it not a barrier, because the reality is we don't have tons of long-term funding to move this forever. So there's going to have to be, in my view, some partnerships, lots of partnerships for people to leverage their expertise in the private sector, other public sectors, in order to get the ball where it needs to be. Thanks. Yeah. Okay. Great question about application timing. And for the RNA interference work, we don't know. We're not to that point yet because we don't know if it's going to be optimal to hit little seedlings before they ever grow, or wait until it gets big enough and then be able to translocate. Or is it going to be optimal on those initial populations where you have a exposed mud flat, or you do a restoration action, and all of a sudden you have frag coming in with those thousands of plants you just planted, in which case you're targeting, species-specific targeting those without killing all the plants that you just did. So that at the early growth stage versus some of the ugly pictures from Western Lake Erie and other places that I've shown, maybe not. I mean, the reality is I think it's going to be a combination of tools will be the best approach through time, depending on the situation. So maybe you have to cut and do an herbicide like a glyphosate or something, and then these other treatments could come into play. So we don't know the timing of the year yet, frequency of application. The bioherbicide, I will say that we have been testing at different times of the year, and I'm not seeing a big difference yet on like the glyphosate, applying fall application, that kind of classic mindset for Phragmites, and that didn't show an improvement in performance compared to earlier in the season. Yeah, potentially. And you may also think that we're thinking about, rather than just spray, also like granular, like again, for the bioherbicide, the microbial work, make it granular or something versus a spray, which then maybe slow release, have it out there, at wintertime or spring, have it there before the plants ever come up. A lot of things to sort out, but I think it's really encouraging to at least want a pathway and try to move this forward. So.
Yep.
Is that right?
Let's give a round of applause.